Now that we have a handle on the structure of a DNA strand and how the double helix comes together in general, we can take a look at DNA replication. Like I said, one of the reasons why we really wanted to know what DNA looked like is because we wanted to know how it worked. We wanted to know how it replicated itself and how the cell could read it to get information. Now, that double helix, that two-stranded system that DNA uses, is actually vital to the way that it replicates its information. Because on a double helix, if one side says A, T, C, G, the other side is guaranteed to say T, A, G, C. Which means that each half of the double helix acts as a carbon copy of the other. It works like a negative image. So in order to duplicate a DNA strand, in order to replicate a DNA strand, what we have to do is break the hydrogen bonds in between the two strands that are holding it together, right? So T binds to A binds to T. There's a little hydrogen bond between the two strands, between the base pairs. We're going to break those apart. We're going to unzip this DNA molecule. And it really is like a zipper. We start at one end and we gradually break those bonds apart in order to separate the two strands. Once the two strands have been separated, enzymes can come in and read the information that's on each strand, and then they automatically know what the missing strand would have said, what it's supposed to say. And in this way, proteins, enzymes that have no understanding of genetics themselves are able to take all the information on those two halves of the DNA molecule, and they're able to replicate a new strand, a compatible strand for each side. DNA replication is what we call semi-conservative. That means that the original parent strand here in blue on this slide, we split it in half and it doesn't disappear. We don't destroy the parent to create the daughter strands of DNA. One half of the original parent DNA is maintained in each daughter DNA strand. So here you can see the idea that we're going to unzip the double helix. We're going to create what's called a replication fork. We're going to split it in half, and right where we're splitting it in half, we're going to fill in some new bases to create the other DNA strand. And when we do that on both strands, at the end of this process, we end up with two complete double helixes that have the exact same information as the original double helix. Another way to say that is that each half of the double helix, each polynucleotide in that structure, acts as a template to make a new strand of DNA, right? We're going to read what's on one in order to figure out what the rest of it should say. Let me give you an example on the board. So, I have a DNA strand here that I've pulled apart. Here's a sugar phosphate backbone, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, going down this side. Here's the same thing on the other side. So the sugar phosphate backbone holds these two strands together, and then hanging off of each sugar, you have a nitrogenous base, an A, T, C, or G. Now you can see that wherever I have a T, on the other side, on the other strand, I have an A to T, T to A to T, C to G, G to C, curvy letters with curvy letters, angular letters with angular letters. And it continues, A to T, T to A to T, and so on and so forth going down this line. Now, once we've unzipped this molecule, once we've broken the hydrogen bonds and separated these two polynucleotides from each other, a different kind of enzyme can come in and it can read what's written on one strand. And it's going to just do what I've been doing in order to make these. It's going to say, oh, there's an A here. I'll make a new strand of DNA with a T in that position. I will make another strand of DNA here. Okay, T, G, C, T. Whenever there's a T, I put an A. Whenever there's a T, I put an A. Whenever there's an A, I put a T. Whenever there's a G, I put a C, and so on. The enzyme can ride along this molecule, making the complementary strand of DNA. Look, just from looking at what's on this one strand here, using this as a template, I have made an identical DNA strand to what we peeled off of it. T, T, G, C, T, A, T, C, right? Going down the line. I can do the same trick on the bottom group here. I can make a second strand attached to this one. I see T, I'm going to put an A. T, A, G, C, C, G, T, A, T, A, T, T, A, C, G. Now there's a sugar phosphate backbone on these new strands as well. I'm not going to write it all out, but here is our sugar phosphate backbone going along the side. Okay. So now, by peeling this away and then using my base pairing rules to create a second strand, I am creating two daughter strands of DNA. Two daughter strands. Okay. Here's number 1, and here's number 2, right? And as I keep peeling the original parent strand of DNA apart, I'm going to expose more and more bases, and then I'll be able to continue my process of making these two complementary strands here. One more time, let me clear away all the extraneous stuff, the sugar phosphate backbones. You should know about the sugar phosphate backbone, but I'm going to simplify the image because this looks a bit of a mess, I understand. If the template strand reads T, A, G, C, T, A, T, T, G, C, C, then the complementary strand, the strand on the other side of these hydrogen bonds, is going to read A, T, C, G, A, T, T, A, C, G, G. These are opposite polynucleotide strands. The individual strands are held together by phosphodiester bonds. That's the sugar phosphate backbone, it's the phosphodiester bonds at work. But the two sets of base pairs are held together by hydrogen bonds. And that is the structure of a DNA molecule. So, when you take one strand of DNA and you make two strands of DNA that are identical from it, it's called DNA replication. DNA replication. Here you can see the blue parent strand of DNA being unzipped. It's unwinding. We're creating a replication fork. We're splitting it in two different directions. Each half of the DNA molecule, each polynucleotide strand, becomes a template for a new strand of DNA to be built. Those new strands are going in orange. Enzymes can read what's on the one DNA strand, and then they can synthesize a compatible strand of DNA to go on the other side. And at the end of this process, you end up with two DNA molecules that are identical to each other. The molecule that actually accomplishes this process of replication, they're called polymerases. There's a couple of different polymerases. Right now I'm just talking about the general idea of polymerases. Polymerases ride along a DNA molecule kind of like a train on a track. They just lock onto it and they ride from one end to the other. And what they're doing is synthesizing DNA. It's an ASE, an enzyme, a polymerase, an enzyme that makes polymers. What kind of polymers? DNA polymerase makes DNA strands. RNA polymerase makes RNA strands. So again, enzymes are named after what they do. These polymerases are very, very good at making new molecules of nucleic acid, whether it be DNA or RNA, which means that they are vital in DNA replication. They're also very handy if you have damage to your DNA molecules. Let's say you go out to the beach and it's a very, very sunny day. There's lots of ultraviolet radiation coming and hitting your skin. That ultraviolet radiation has enough energy to damage the DNA inside your skin cells. This is why not wearing sunscreen can increase your rate for skin cancer. But these polymerases, certain kinds of polymerases, can come in and they can find the broken or missing parts of your DNA strand. They can find the location where certain bases have been knocked out by ultraviolet radiation, and then they can put the correct base back in and repair that DNA. So they're useful for a couple of different things inside the cell. Now, we're going to cover a few different processes related to DNA and information processing and proteins and this sort of thing. And in order to keep track of these processes, I'm going to teach you what's called the central dogma of molecular biology. I believe that term was actually coined by James Watson of the double helix fame, but it's a pretty common phrase. You'll see it in a lot of biology textbooks. Any biologist you go to, you say central dogma, and they'll know exactly what it is that you are talking about. The DNA is the information carrier of your cells. It's the information carrier of you. It's what determines what kinds of proteins are going to be made. So DNA holds the information, but the proteins are what actually do the work. They're the molecules that will actually cause your traits to occur. So if you are very, very tall, it's coded for in your DNA, but it's the reason why you are tall is because of the activity of enzymes over the entire course of your development, going from little baby on up to full-grown adult, right? So the DNA is the information holder, but proteins are what actually gets the job done. So in order to get traits, we need a way to go from DNA to proteins. And these two molecules don't speak the same language. We need an intermediary here. We need something to go in between the two of them that's able to carry information from DNA, which is locked away inside the nucleus and not allowed to leave except that one time during cell division. The DNA is not allowed to leave, but we need to get the information to the ribosomes so they can make proteins. How are we going to accomplish that? We need another kind of molecule, another kind of nucleic acid, and it's called RNA. Okay. Now, here's the central dogma of molecular biology. If you want to take a DNA molecule and you want to use it to make more DNA, that's called replication. DNA replication. DNA can make more DNA. It's very good at copying itself because it's two-stranded. If you want to actually use the DNA, the first thing you got to do is copy the information in the DNA over to RNA, and that's called transcription. Transcription is taking DNA information and putting it into RNA information, copying it down. The DNA, again, is not allowed to leave the nucleus, but RNA, if you make a copy of the information in RNA, that is allowed to leave the nucleus. So the RNA is like a photocopy. I think of the nucleus of the cell as being kind of like an archive of all the blueprints of proteins that the cell might decide to make at some point, and you're not allowed to leave with any of the original blueprints, but you are allowed to make photocopies and take those photocopies out of the archive. RNA is that photocopy. And when we copy DNA information into RNA, it's called transcription. And then to actually make a protein, we need to shift languages. We've been in nucleic acid language, but we need to convert over to protein language, to polypeptide language, to amino acid language. That process is called translation. Translation is taking information from RNA and putting it into proteins. We're going to talk about every single one of these processes and all of the enzymes that are involved that make these processes possible. But this is called the central dogma. The central dogma of molecular biology. DNA gets transcribed into RNA, and then RNA is translated into proteins, and then proteins give us all of our traits. They do all of the work of our cells. They're the things that actually get things done.